Hello, students. Very good evening. How are you all? Had good cup of tea, coffee already. Had a busy schedule in the college. Had a lot of assignments from the college. Some exam time is going on for some. COVID time is also going with us. Many things are up to. Are you having a regular exercise or mental health things? Because with this COVID thing, our mental health is very important, which we never discussed before. It was never discussed in our culture, and it has been discussed in the West quite long before, because they have a lot of mental problems. But in India, we never had. We have already such a busy schedule that we never thought of these things. So just take care of your ones, near ones, dear ones, the old people, how they are going through, and please make sure that everyone is fine. Talk to them, just ask how's their health, how's their work, how's their daily routines looks like, and just talk to your friends. Maybe talking is a good therapy, and sharing your things, your inner energy, and bring it to the vibe out to your close friends, and sharing each and everything what you're going through. It helps a lot. It helps a lot to keep a balance, especially during this COVID time when we are supposed to stay at home, and it's very hard to maintain a good relationship. In a good relationship with your inner dear and near ones. So keep tracking. So that was something about today's Gyan. Any questions you have, students? Before we start, it's always good to have some Q&A fire, so that we can start at a good level all together. Anything about spirituality, meditation, exercise, maintaining your body health routine, how to remember many things within less span of time, how to be good in your education, how to have clear thoughts, how to maintain good confidence level like serotonin, dopamine, how to maintain their hormones level balance in our body, not to have them too much, not to have little less also, so that we are always in high energy and can do maximum things within a short span of time. Anything else, anything related to that, you can feel free ask to me even personally. um, okay, if not, then we can start our session today. So today, in today's session, we will be dealing with chip, that is chromatin immunoprecipitation. We will be dealing with the blotting techniques, and if we still have time, we will go for pymol. So before we go ahead, please, I will recommend download this software pymol. If you have Windows, so you can download it from here. This is for the people who are studying from laptop at the moment. In the from the mobile who are studying, it's not possible. So you can download this. So this software will help us to know about the structures of proteins, how you can manage them, how you can do it. So we watch a small video for the same, and then we go for it. So while we are studying, I can give you 5 minutes. You can download that software and start having a rule, even though I don't have at the moment, but some weeks I have 5 a.m. 5 a.m. rule. It's a famous book by Robin Sharma. So he says that when you wake up 5 in the morning, first what you do is you do some exercise for 15 to 30 minutes, then you go for your meditations, and you start doing the utmost important tasks, like to make a do list of your morning work, and then you started to sort out things as per as you want to do it. So you can do many things by doing so, and maintain a very high energy level throughout whole day. And by the 7 or by 8 you should have your dinner for sure, and then by 9 you're in the bed to wake up early in the morning again, 9 or 10. So 7 hours of sleep is enough to have a sound to recharge our body, to keep our hormones in balance again. So today is day 7. Good evening, students. Good evening. Very good evening. So we can start, I guess. So chromatin immunoprecipitation and its principles. Today's day, it was from the day yesterday, but let's do it today. So in this case, in the chromatin immunoprecipitations, you basically check DNA and protein interactions. Which part of DNA being interacted with the protein or the transcription factor while they are having interaction. So what you do is you cross-link your cells with the DNA, and then you lyse them. Then after lysing, you have antibody binding to it. Then you do the immunoprecipitation. Then you have a wash steps. You remove the antibody. Then you do the reversible cross-link, that you just extract DNA out of it, that which has been attached to your protein. Then DNA purification and quantitative PCR is done, and then DNA and protein analysis is done afterwards. So that's the basic principle of chromatin immunoprecipitation. CHIP, it's also called CHIP. So you might have heard these terms: CHIP sequencing, CHIP RNA sequencing, and all these terms. So what happen in this? Not only protein interact with each other, but they also interact with DNA. And CHIP is a technique that determines whether the protein of interest interacts with a specific DNA sequence. So it's find out the interaction between your DNA and specific DNA sequences that are required to be worked out. And this technique is often used to study the repertoire of sites of DNA that are bound by particular transcription factor or by histone proteins. So we also study transcription factor or histone proteins how they are interacting with the DNA, and looking at the precise genomic equation of locations of histone modifications. So their histones are modified. So you might have heard these beads like strings like structures, including acetylation, phosphorylations, or methylation. So all these things are being modified with the help of acetylation, phosphorylation, methylation in histones, and we can find out with the help of chip immunoprecipitation. So how does the chip works actually? So chip can be used to determine the presence of protein-protein interaction, protein-DNA interaction at steady states, to quantify the changes in the interaction at specific phases of cell cycle or following the treatment of interest. So what we do is, protein associated with a chromatin are temporarily cross-linked first in the live cells or in the tissues. So we use formaldehyde or UV light to do so. Then we shear them with the help of enzymic digestion, or what we can do is we can do the sonifications to yield fragmentations of a DNA around 300 to 1000 base pairs. A protein of interest along any associated DNA fragments is immunoprecipitated then from the cell's debris using a specific antibody. The cross-link is then reversed and DNA fragments are purified. Whatever the cross-links that were there, they are reversed and DNA fragments are purified, and these DNA fragments that we alluded, they went through the quantitative real-time PCR, QRT-PCR, using primer flanking the genomic locus of interest. So with a particular DNA, we flank that part, and with the help of that, we increase its production. Then DNA amplification is done as indication of enrichment in the binding of the protein interest. So DNA amplified of the indicated regions, and that region will be known as the protein of interest that was binding to the DNA. So this way we can find that region actually. So there are some modified chip techniques beyond this. So according to that you can work. So the chip on chip technique. In the chip on chip technique, there is a genome wide analysis of protein binding sites using microarray array analysis of purified DNA. Just a second, students. Hello. G. थैंक यू सर, आई एम बिट बिजी एट द मोमेंट. नहीं सर, अभी ज़रूरत नहीं है सर. थैंक यू सर. Sorry. So there is chip on chip where you do the genome wide analysis of protein binding sites using microanalysis of purified DNA fragments. Then there is chip sequence. In this genome wide analysis of protein binding sites, using, instead of microarray, we use deep sequencing of purified DNA fragments. So we go for deep sequencing also. Native chip, in this we omit the cross-linking step and use micrococal nucleus digestion to cut DNA at histone linkers to examine the DNA target of histone modifying proteins. So instead of using the cross-linking, we use a digestion, nucleus digestions, to cut DNA at the histone linkers to examine the DNA target of histone modifying proteins at the end. Then chip exo, it's the addition of exonuclease digestion steps to obtain the increased resolution of protein binding sites up to single base pairs. Chia PET, so it's a chromatin interaction analysis by paired and tag sequencing. So in this we combine the principle of chip with the chromosome conformation capture to detect the long chain chromatin interactions mediated by a protein of interest. Then there is ICHAP, indexing first chromatin immunoprecipitation. This is a high sensitivity technique that reduces the number of cells required for chip experiments by initially barcoding total cellular chromatin. So initially we just calculate the total cellular chromatin by that, so our cell requirement is less. Then there is an NCHIP. This is engineered DNA binding molecular mediated chromatin immunoprecipitations. And this we employ the CRISPR-Cas9 that we have discussed in previous lectures in detailed manner. And then RIP chip, RIP sequence. In this one, it's similar technique. Instead of DNA, we check the protein RNA interaction in the RIP chip. So there are some limitations of chip. It's not all the techniques are all good. So they often have low yield signals as compared with the controls, leading to the inconclusive data that is limited to the resolutions related to the size of DNA fragments, which make it too difficult to determine the exact binding site of a protein. While chip will infer the presence of protein at a given genomic locus, it cannot determine the functional significance of the protein binding at the DNA region. Cross-linking may be additionally include proteins that transiently interact with the DNA or DNA binding proteins have no functional significance. Similarly, DNA interaction of proteins with short residence time may not be fully captured. Additionally, interacting proteins may mask the epitope of the protein of interest. So some important factors to consider, I will go through the last part, that is to maintain appropriate controls. That is very important. When you are doing experiments, you have to maintain positive controls, negative controls, input control, IgG controls when you are doing any experiments actually. As for any experiments, controls are indispensable in chip to regulate enrichment in protein binding and to interpret the significance of results. So following controls are recommended. So you can have isotap matched control immunoglobulins. In this, the appropriate non-specific IgG is added instead of protein-specific antibody at the same concentrations. Input control to save 5 to 10% of cell life before the addition to normalize for variation in starting material between consecutive experiments. Positive control locus, if possible, a DNA regions where your protein of interest is known to be bind should be examined, and this will tell you if a chip experiment worked. Negative control locus, if possible, a DNA regions where protein of interest is not expected to bind and should also be tested. This will tell you that your chip experiment is specific. So we have discussed a little bit about chip, how chip is working, what are the various modifications you can do, what are the limitations, what are the controls that you have to maintain. Now comes a little bit of summary of that.
Come in closer, let me whisper something in your ear about ChipSeek. Yeah, StatQuest. Yeah. Hello, I'm Josh Starmer and welcome to StatQuest. Today we have a gentle introduction to ChipSeek. Note, this StatQuest is based on the gentle introduction to RNA-seq. So watch that first unless you're already totally down with RNA-seq. Okay, we've got a bunch of cells. Inside each cell, there's a nucleus. And inside each nucleus, there are a bunch of chromosomes. Let's focus on the chromosomes. Specifically, let's focus on these chromosomes. Chromosomes are made out of chromatin. Chromatin is made out of DNA, plus histones, a type of protein, plus other proteins that we'll talk about some other day. They're not that important for understanding ChipSeek. DNA wraps around the histones to package DNA, and the packaging can regulate gene transcription. Depending on how they are modified, histones can activate or repress genes. Lots more can be said about chromatin and how it's packaged, but that's for another day. Today we just need to know that chromatin is essentially DNA wrapped around histones. To simplify things, Let's just use this big blue line to represent chromatin from here on out, but remembering that it's made of DNA and histone proteins. And let these big brown arrows represent genes. And let this green circle represent a histone that allows transcription. And let this red stop sign represent a histone that represses transcription. In a cell, all kinds of proteins can bind to DNA. This mustard-colored thing might promote gene transcription of this gene. And this pinkish-colored thing might repress this gene. And who knows what this green thing is doing? We can use ChIP-seq to find out. ChIP-seq stands for chromatin immunoprecipitation combined with high-throughput sequencing. It identifies the locations in the genome bound by proteins. That's the most important thing in this stat quest, so I'll say it again, in bold. ChIP-seq identifies the locations in the genome bound by proteins. For example, say like we wanted to identify all the regions in the genome bound by the green thing. The first thing we do is use formaldehyde, or something like it, to glue all the proteins bound to the DNA together with the DNA. This means that all of the DNA-bound proteins, including the ones we're not interested in, are glued to the DNA. The next thing we do is cut the DNA up into small, approximately 300 base pair, fragments. Then we isolate the protein we're interested in this case, we're interested in the green thing, using an antibody. The black star represents an antibody that is attached to a bead. Then we isolate the proteins bound by the antibody and wash everything else away. Then we reverse the formaldehyde glue by warming up everything. Then we isolate the DNA by washing away the proteins, including the histones. Now that we see how to isolate DNA that is bound by a particular protein, let's take a step back. So far, the example has focused on just these chromosomes. But the process, glue proteins to DNA, cut up DNA, bind proteins of interest with antibodies, isolate antibodies, unglue and wash away proteins, applies to all the chromosomes in the cell. And it is usually applied to a pool of 6 million cells, give or take a few. So we end up with a lot of DNA fragments from a lot of cells. Then, just like with RNA-seq, we prepare a sequencing library by adding sequencing adapters to both ends of the DNA fragments. Then, just like RNA-seq, we PCR amplify the library, check the library concentration, sequence, filter out garbage reads, and then align the high-quality reads to a genome. That is to say, if this is the genome, the first read might come from here, a location on chromosome 2. The second read might come from here, a location on chromosome 1. The third read might come from here, another position on chromosome 1, etc., etc., etc. Ultimately, we get a long list of genomic coordinates for all the reads, usually between 50 and 100 million reads. And we can use those reads to create a genome browser track. These are genes and chromosome positions in the mouse genome, MM10 to be exact. This is the track that we created for our ChIP-seq reads. A lot of reads map to this region, and relatively few reads map to these other regions. This track was made from a control experiment. The control track was made by taking some of the input chromatin from the original ChIP-seq experiment, and, without using an antibody to enrich for any particular protein, ungluing all the proteins and washing them off. Then sequencing, aligning, and converting into a track. In summary, The control track uses some of the same input chromatin for the ChIP-seq experiment, but doesn't try to enrich for any particular protein binding. We use the control track to verify that a high concentration of reads in the ChIP-seq track is due to a protein binding there, and not because a lot of reads map to a repetitive region. Statistically significant peaks are usually represented on genome tracks by rectangular bars. We could then compare peaks for the same protein in different cell types, like lung versus kidney. Or, if we didn't know the specific DNA sequence that this green thing bound to, we could guess that it is a motif found in all of the peaks. Here's a motif found within the peaks that indicate where the green thing bound. The large letters are more frequently associated with the green thing than the little letters. We can also try to determine the functional role of the green thing by looking at where it binds relative to the genes. Here, we see that the green thing binds near the start of a gene, so it might regulate that gene in some way. Anyway, those are some of the things you can do with ChIP-seq. In summary, ChIP-seq identifies the locations in the genome bound by proteins. Bam! Hooray! We made it to the end of another exciting stat quest. If you like this stat quest and want to see more, please subscribe. To support StatQuest, please click the like button and consider buying one of my original songs. Okay, until next time, quest on.
So that was quite nice summary of how from start to end of your chip sequencing, where we have discussed how to interpret data, like the protein has been, they have found that they have been it's at the promoter region, so something to initiating the DNA resolution. So basically DNA chip sequence is done to check the identify the protein and DNA interaction, and that's what we have discussed today in this lecture. So thank you very much for this lecture, for being there. So any questions, students, by so far? So that we can start with the next part. So I can share this chip sequence with you. Yeah, by 20th. By 20th you have to submit it. Okay, so we go for the next part then. Blotting technique. So we were discussing yesterday about electrophoresis. We discussed quite details, many zone and molecular sieving techniques, many other different types of electrophoresis. So today we're gonna discuss about MBBT in this part, blotting techniques. So mainly we will discuss in this is southern blotting, northern blotting, western blotting, northern western blotting, southern western blotting. So mainly the southern blotting is done to check the DNA part. Northern blotting is to check RNA, whereas the western is done to check protein. And northern western would be then RNA and protein interaction. Southern western would be the DNA and protein interaction. So that's how all these techniques are differentiated. So this is the appendix. We will discuss southern, northern, western, then southern western, northern western, then dot blotting and zoo blotting also. So first is southern blotting. So southern blotting basically is a hybridization which detects target DNA fragments that have size fraction by the gel electrophoresis. And this technique was invented in 1975 by E.M. Southern. The basic principle is that you exploit the property of radio level probe with a single stranded DNA. If we want to detect the presence of a specific sequence in our mixed DNA samples, then we accordingly design the probes which have complementary sequence to our target sequence. So in this procedure called southern blot, DNA from the sample is cleaved into restriction fragments with a restriction endonucleus, and the fragments are spread by the gel electrophoresis. So here you can see you have your gel run with the help of electrophoresis in the first step using the radioactive markers as size guide in the first lane. So you have a labeled actively markers specific size and this is electrophoretic gel. Then you transfer your samples to the nitrocellulose membrane by keeping a stack of paper towels on top. Then further, these papers are nitrocellulose papers now contain nucleic acid print, and gel is copied faithfully blotted onto the nitrocellulose. Then you put them into the radioactively labeled nucleic acids, uh, and then they could be rinsed. So photographic film is laid over the paper and exposed only in the areas that contain radioactivity. Nitrocellulose is examined by radioactive bands indicating a hybridization of original nucleic acids with radioactive labeled ions. So here your with the ones which are radioactively labeled, they will be hybridized with the nucleic acids, and you can see your results like this. So you can have a lot of information from southern blotting, whether a particular gene is present, how many copies are present in the genome of an organism, the degree of similarity between the chromosomal gene and the probe sequence, whether the rearrangement have occurred during the cloning process. So all these things could be checked out by this technique. So uses of southern blotting, it could be used to detect cancer cell genetic diseases such as monoclonal leukemia, sickle cell mutations. We could do DNA fingerprinting, genetic engineering, forensic science for paternity testing, personal identification, sex determination, species exclusions. You can identify related DNA sequence in the genome to determine if there is a gene family and to determine the copies of particular DNA sequence presented in the genome of organism. Identify gene mutations, deletion, duplication, gene arrangement involved in the disease. So let's go through this. Southern blotting technique. So you load your sample first in the TB gel. Then you add denaturing solutions, NOH and NaCl. Add neutralizing solution. Then you do the southern transfer. Soak the paper in transfer buffer 10x SSC. You put your gel, then put nano membrane on the top. You cover it with the plastic wrap. And some weight on top. So you rinse your nano membrane in 6x SSC, and then place the membrane on the UV translumimeter to check the crosslink. Then you do the hybridization method. So you had pre-hybridization buffer into a big uh, revolving tube. You add your nylon membrane into that. Then incubate at 42 degrees Celsius for 2 to 4 hours. Then again add pre-hybridization buffer. Then isotopes, add isotopes labeled DCTPs, which will bind to your membrane into the hybridization buffer. So basically hybridize means here getting attached, getting joined together, hybridizing. So your your DNA, yes, is getting hybridized to your membrane, nylon membrane. Wash it with the wash buffer to remove any extra rinse. Incubate at 52 degrees Celsius for 30 minutes. Now it's ready to expose. We can see under X-ray film ray our membrane and could look for the data. That was the method how you do the southern blotting. Now comes the northern blotting. Instead of DNA here, simply it's RNA is there. So just have to check instead of DNA. Any questions so far? Yeah, as I discussed, so it's helping in pre. So hybridization could not be done at one level. So first have to be done at the lower level with the pre-hybridization thing. Then you can do it with that. Okay, you can do it by 25th also, if it is told like that. So in this case, in the northern blotting, instead of DNA, we check for RNA. So RNA is extracted from the cell of interest. Precautions has to be taken care very much in this case, because of your skin and glasswares, that you all body have RNA, so you can get mixed with your RNA could be depleted out. Wear gloves with specifically treated plastics, glasswares to avoid accidentally introducing ribonuclease to extraction step. Addition of diethyl pyrocarbonate deep depths inhibits ribonuclease activity, and also baking at high temperature destroys ribonuclease activity. So all these things have to be taken care. So in the northern blotting, RNA is isolated from the several biological samples. RNA samples are separated according to the size of enclosed gels. The gel is then blotted on the nylon membrane or nitrocellulose filter. The membrane is placed in a dish containing hybridization buffer with a labeled probe. Membrane is washed. It's the same method like what we discussed before. Instead of that, you have RNA in this case. So this technique could be used to check all the applications which are related to RNA, such as to researchers could determine the gene expression patterns. They could detect cancerous pancreatic cells, tissues. It could help to know the functions of proteins. It could help the scientists to detect the size of RNA, to alternate splice products using the process of partial homology. So let's continue with the northern blotting. So the RNA, formamide, formaldehyde, and MOPS. So these are the, to prepare your RNA sample, you add all these things to your append of. It's again same method again. You soak your Wartman paper, filter paper. So all process was same. Instead of DNA, we have RNA in this case. Procedure remains the same. Just have little bit of change of buffers here and there, but things were same, quite same. So which technique? So it depends. All techniques are important. Yes, Western blotting to check between your disease state. You have lymphoma cancer cells, some melanoma cancer cells. You check there straightforwardly for the Western blotting protein expression, that what is the disease state and normal state, and you check for the genes which are cancerous cells have, and then you can make a differentiate that that person has cancer or not. Whether it's a heart patient, you do the biopsy of that person, or in the liver side get the liver tissue via biopsy, and then you can check with the help of that whether you want to check at the DNA level, RNA level, or protein level. It depends which technique you want to go for. So all your questions. So but these things are done as radioactively labeled, so they will put some X-ray rays on top to check your membrane, nylon membrane. So that's why they are being closed so much from up and side down. You have seen also that there is some shield in front of the technicians, so that they should not get uh, these any harmful effect from these rays of the radioactivity. So all these things have been taken care while doing these experiments. So yeah, most important to me in my case, I use mass spectrometry. uh, Mass spectrometry and LCMS, liquid chromatography, immunoprecipitation, gene cloning, um, daily routine of cell culture, microscopy, and yeah, that's basic. You have to count your cells, you have to make primary cell culture, secondary cell culture. It depends upon what is you're looking for. So all any lab that you go, they will be specialists for one technique and allied techniques that they needed. So it depends upon which lab you go and start working there. So, and students, which is your favorite lab technique so far you have studied here? Could you share here in the comment box? I would like to know. Which is your favorite lab technique so far you have done by your own hands, or you have studied here with me? Which is your favorite so far? PCR, yay. CRISPR Cas9, okay. Gel electrophoresis, yeah, that's also my favorite. I did a lot of them in my life. Western blotting, animal cell culture, yes. RT-PCR, yeah, it's also very important. When you want to analyze things at the mRNA level, then you can confirm things at the DNA, at the protein level. So you will get a chance to work with CRISPR also if you got to work in somewhere. So when I was doing my PhD in 2016, CRISPR was, people were doing that CRISPR thing, and they published really good papers with that. FRET, wow, fluorescence. I one of the biggest scientists was doing that FRET technique there in Germany. Okay, super awesome. Then we go for the Western blotting now. Western blotting technique is an analytical technique used to detect specific proteins in a given sample of tissue, homogenates or extract. Sometimes referred to as immune blotting, and this technique uses gel electrophoresis to separate native or denatured proteins by the length of polypeptides. So the basic principle is electrophorized on an SDS page and electrotransferred onto the nitrocellulose membrane. The nitrotransfer membrane is detected using specific primary antibody and secondary enzyme labeled antibody and substrates. So in this technique, first we have a sample of protein which is separated on the basis of the molecular mass using SDS page. And then electrophoresis moves the protein from the gel and onto the nitrocellulose membrane where the proteins adhere. To detect the protein specific protein, an antibody to that protein must be available. A nitrocellulose membrane itself has a nitrocellulose membrane itself has many non-specific sites that can bind proteins and include antibodies which must be blocked with the non-specific protein solutions. Then milk is added to the primary antibody to the protein of interest. The antibody mixture is detected using the secondary antibody that has label attached to it. Often a reporter enzyme such as alkaline phosphatase is linked to the secondary antibody, and addition of X-phos or to the blood allow to check the protein band. So you have a nitrocellulose membrane with attached protein to it. Then you add non-specific proteins and primary antibody. And primary antibody attached to the specific protein that you are interested for. Then secondary antibody binds to the secondary to primary antibody. Then with the help of X-phos or with the chemical luminescence substances, you can calculate the secondary antibody. So there is information that you obtain from the Western blotting, that is size of protein and expression amount of protein. Its uses are very diagnostic techniques. You can use Western blotting, widely used. A positive Western blot technique can usually confirm HIV infection, and also for definitive tests for bovine spongiform encephalopathy, BSC, mad cow disease, and also diagnosis of some form of Lyme disease also. So let's go through in thorough detail about Western blotting technique.
In this video, you will learn how to perform a Western blot. A Western blot can be used to identify specific proteins in a sample and provide information about the protein size and relative abundance in the sample. First, fill a tray with blotting buffer. You will be using this buffer to equilibrate your gel prior to starting the Western blot. Next, remove the gel from the gel cassette using the opening key. Line up the arrows on the opening lever with the 4 arrows on the cassette to open the cassette. After trimming the top and bottom of the gel with a straight edge, equilibrate the gel in the tray with blotting buffer for 15 minutes on a rocking platform. Pre-soak fiber pads in blotting buffer so that they are thoroughly soaked. To make a blotting sandwich, obtain a container large enough to fit the gel holder and add enough blotting buffer until the container is filled approximately 1 centimeter deep. Place the gel holder cassette in the container with the black side down and immersed in the buffer, and the white side up and out of the buffer. Lay 1 fiber pad flat on the black plastic. Next, wet a piece of blotting paper and place it on top of the pad. Be careful to avoid any bubbles between the pad and the paper, and make certain the buffer covers the paper. Take the gel and carefully place it squarely onto the blotting paper. Again, being careful to avoid any bubbles between the gel and the blotting paper. Next, you will be applying a piece of nitrocellulose membrane. Remove the protective sheet from the membrane and wet the membrane with blotting buffer. Carefully place the membrane squarely on the gel. Avoid moving the membrane once placed on the gel, as proteins will begin to blot immediately. Using a roller, remove any air bubbles between the gel and the membrane. Place a second sheet of wet blotting paper on top of the nitrocellulose membrane. Place a second wet fiber pad on top of the blotting paper. Fold the clear plastic side of the gel holder over the sandwich and clamp it to the black plastic side by sliding over the white clip. This tight fit will squeeze the sandwich together. Insert the gel holder into the inner module. Make certain that the black side of the gel holder is next to the black side of the module. Place the inner module into the electrophoresis chamber. Add a frozen cooling unit and fill the chamber with blotting buffer to the level of the white clip on the gel holder. Place the lid on the electrophoresis chamber. Connect the electrical leads to the power supply, making sure the connections are correct, red to red and black to black. Turn on the power supply and run the blot at 20 volts. If a timer is available, set it for 2.5 hours. When the run is complete, turn off the power supply and disassemble the electrophoresis chamber and remove the inner module. Open the module and place it in a container filled with blotting buffer with the black side down. Starting with the first fiber pad, remove each layer until you reach the nitrocellulose membrane. As you remove the membrane, note that the proteins have been transferred from the gel to the membrane. Note that the kaleidoscope pre-stain standards have been transferred and can be seen on both sides of the membrane. You can also see that there are no longer any proteins on the gel. Immerse the membrane in 25 milliliters of blocking solution and incubate it for 15 minutes at room temperature on a rocking platform. Pour off the blocking solution. Add 10 milliliters of primary antibody. Incubate for 10 to 20 minutes on a rocking platform. The platform should be set at a faster setting to ensure constant coverage of the membrane. Pour off the primary antibody. Rinse the membrane quickly in 50 milliliters of wash buffer and then discard the wash buffer. Add another 50 milliliters of wash buffer to the membrane and let it wash for 3 minutes on the rocking platform at a medium speed setting. Discard the wash buffer. Add 10 milliliters of secondary antibody and incubate the membrane for 5 to 15 minutes on the rocking platform at a fast speed. Pour off the secondary antibody. Rinse the membrane quickly in 50 milliliters of wash buffer and discard the wash buffer. Add an additional 50 milliliters of wash buffer and wash the membrane for 3 minutes on the rocking platform on a medium speed setting. Discard the wash buffer. Add 10 milliliters of substrate. Incubate the membrane with the substrate for 10 to 30 minutes with either manual shaking or on a rocking platform. Watch for the color development. Once the colors have developed, rinse the membrane twice with distilled water and blot dry with a paper towel. Air dry for 3 to 60 minutes and then cover in plastic wrap for storage.
So this is how your results looks like. So you have your molecular marker here and your gel running with different samples and your proteins coming out like this. So this is how the end results looks like. So any questions? Oh, I'm flattered. Thank you so much. Frozen cooling unit, to let that transfer to be done at the cool temperature, the transfer is done very well. So that's why we keep the frozen cooling unit. That's the main reason. Okay. Mine favorite is all. As much in my life I could learn more and could teach more. It's all good for me. In this case, they didn't tell me the technique that we have gone through. What is the end result signifies? But as we discussed yesterday, I did many experiments with uh, ER stress markers like RE1 alpha and PERK, and I added tunicamycin, thapsigargin, H2O2 DDT. So I see significant changes in the CTGF, my CTGF protein. So these results were found in the Western blotting in the protein level. So that's what you are looking for. What is your hypothesis is, and your hypothesis is doing with the methods. In the methods, if your Western blot results are coming out to be true, so that will be a nice results for your explanation. So just be careful with your hypothesis, what you're looking for. Sometimes what you're thinking is something opposite is happening, and you're always thinking that, oh, I was thinking this and this was happening inside the cells. So this happens many times. So our hypothesis could also get wrong. In my case, my paper was retracted. So somebody already, not retracted, but somebody already published it before me with different story. So my results doesn't matter then at the moment. So I have to improvise my results so that they are opposite of what have been published. So that's what I did in my PhD. So I worked for 3 years hard, but somebody already published my work. Then I have to publish in a way that people would love to know my story also. So it's not just hard work wasted out. So I still published it. It doesn't get that recognition that the first paper has, because first paper is always known in the market than the second one. So this happens sometimes. So southern western blotting is dealing with and to know the interaction between DNA binding proteins. In this mixture of proteins such as crude nuclear extracts or partially purified proteins are first fractionated on a sodium dodecyl sulfates, and the gel is then equilibrated in the SDS buffer to remove detergents, and then protein transferred by electron blotting to an immobilizing membrane. And during the transfer of proteins, renature and hence DNA binding proteins may subsequently be detected on the membrane by the ability to bind labeled proteins, DNA labeled proteins. Northern Western is to check the interaction between protein RNA interaction, which are detected as a hotspot on the filter. Dot blot technique is a modified version of the Western blot technique, in which we identify and analyze protein of interest. Dot blot methodology differs from the traditional Western blot technique by not separating protein samples using electrophoresis. Sample proteins are instead spotted on the membranes and hybridized with an antibody probe. Then comes the last technique of today, that is zoo blotting technique, and it is based on the principle of southern blotting. In the genome of any organisms, there are two regions, the coding and non-coding region. It is the coding region that causes the cause of interest for most of the researchers, as it is associated with the genetic information of specimen. But most of the protein is also non-coding, and question is how to identify coding region in the large amount of the non-coding DNA. So zoo plotting is precisely helping us to distinguish between coding and non-coding regions. So during the evolution, the base sequence of non-coding DNA mutates and changes rapidly, where the coding sequence changes much more slowly. So therefore, in DNA is extracted from a series of labeled animals, related animals such as human, monkey, mouse, hamster, and sample in these DNA zoos are cut off with the suitable restriction enzymes, and they run on the gel and transferred to the nylon membrane, and they are probed with the DNA that is suspected of being human coding DNA. The DNA sample really does include a coding sequence. It will probably hybridize with some fragments of DNA from most of the closely related animals. So here you can see that on the left side is the non-coding bind to itself. On the right hand side you have rat, cow, dog, human, and the probe is coding DNA all species having related sequence. So probe, in this technique, probe is segment of the human DNA that may or may not be from the coding regions. Blotting is carried out as usual. On the left, only the hybrid seen was between the probe and human DNA. So in this hybridization, uh, we see the hybridization with the non-coding stuff there. Therefore, sequence were not found in other species. So it was not found in other species, and probably is a non-coding DNA. On the right hand side, the probe binds to the related sequence in other animals. Therefore, this species DNA is probably from the coding regions. In this region, when they are doing it, so they are being coded for this. So these regions are the coded regions of other species also, of the same species. So this is the non-coding region, and this is the coding region of human, which has been found in other organisms also. That's why it's called as zoo blotting. So that's it. This zoo blotting is not used much, but mainly the western blotting, sometimes southern blotting, western blotting, northern blottings are also used. But once you check things at the protein level, so most of the things will be done easily in that case. So we still have some time. So did you already, students, have Pymol software in your laptop? If not, if yes or no, I will share the video with you, all of you, so we can go through the video and just try to stimulate at your home with that software. This is Pymol 1. So now we are looking for how to check your protein structure at home. Rasmol is also fine. It's the old version. Pymol is much better. So whatever the video is saying you, please go through that and keep replicating in your house also. So even I can do it. So this is your interference of your Pymol software looks like. So I will not speak much now.
Macromolecular models are complex.
So I stopped doing it because I thought that will be interfering with your things. So I hope it was clear, exciting thing actually. I also really like this. You can move your particles like this anyway. So these are 4 heme groups are being there. You can see I made a rainbow color, 4 different polypeptides, and you can zoom in, zoom out. So you can do many things with it, many things. So I just share with you everything now so that we don't forget. Also share one English protocol of Western blot that you might be in need of. So all are being shared now, all videos, everything. So commands for the Rasmol and Pymol similar. I'm not sure. Maybe it's similar, but Rasmol I know is the previous version of Pymol. I mean, it's not a previous version, but it's a older person. Even Pymol is very old. No one using Pymol nowadays. But in my PhD, I used all Pymol, and it was very useful for some scientists. It's really good still now, and it's working well. So it was a basic to teach you about how you can check the various protein structures, and you can play around to check whether what you have to do to look for the post-translational modifications or anything. So that's it. Any questions, students, so far? So we are done for today. If not, um, I thank you everyone for being there, and we see us at the same time. Tomorrow we will do the facts analysis, flow cytometry. There are other softwares. I mean, Pymol is still the number one if you are into the protein structures. So Pymol alternatives. So you can go through these softwares: Rastop, Qtmol, Rasmol, Jmol, BioBlender, UCF Camera. So these are all alternatives to it. Jmol is also used a lot. VMD is also being used. Visual molecular dynamics. Rastop, Calistry. So there are the various alternatives to it. Just play around. You can download these softwares on your house. Just get one protein and go onto the PDB website. There also you can learn a lot. Protein database bank. There you can also check the softwares of your concerned proteins. You can check with the coronavirus what are the structural view of coronavirus, how it looks like. It's nothing coming out about that. Ca- Capsicum receptor TRP1. So this is October molecular of the month. So by this you can look around many things. Okay, students. If you have no further questions, that is for today. I see you tomorrow, same time, same place, and thank you very much. Bye.